See, our colloquia here are kind of an adventure of some sort. So I'm very, in fact, I'm on the injured reserve this year. Sorry, faculty. So all I can do is eat. So I'm really pleased to echo what Josie said to introduce Nils Volkenborn, who's now at the University of South Carolina. And he's really indicative of the sort of new group of young researchers with an interdisciplinary approach to looking at benthic processes and looking inside sediments, which look so innocuous when you walk over them or swim over them. But looking inside, you can see just how exciting they are and why people like me have been interested in them, and Glenn and Josie and others here are so interested in them. And he's going to actually show you why they're so interesting using a wide array of newly applied sensor technology. It's very exciting. Now, Nils did his undergraduate and graduate work in Germany at a range of institutions that I will not attempt to pronounce. He got his PhD at the University of Bremen in Germany, but also worked with the MPI and the Alfred Wegener Institute in Germany as well. So he has background both in a more traditional biological area, in zoology and benthic ecology, and as you'll see, he's moved with great enthusiasm into the quantitative world of biogeochemistry and sensor work. So we're very pleased to welcome you, Nils, and looking forward to your presentation. Thank you.
So obviously, Josie, Bob, I want to thank you very much for inviting me. It's a really great honor being here, meeting already several people here at a place where so much knowledge is floating around. I'm really glad to be here. Today, I want to talk about geochemical dynamics in bioturbated sediment, and also, or especially in this topic, there's so much experience here at this university, but I will try to give my best to add something to it. First of all, I would like to acknowledge my co-authors, especially Lubos Polarewski, who is at the MPI, Max Planck Institute in Bremen. He not only introduced me into the planar opto imaging of oxygen, he has also been, since, 6-7 years, a really great collaborator with lots of forth and back between, now Germany and the US. David and Sally, both professors at the USC in South Carolina. 4 years ago, actually in 3 days, it's our 4th birthday, that they brought me over. Obviously, I need also to thank them, and it's also a lot of fun working with them, and I learned a lot. So why do we care about the sediments? Now, this is basically what you probably would sample if you just randomly would sample a square meter of seafloor, square meter of earth, just randomly, because by far, marine sediments are the largest ecosystem on Earth in area coverage. And so already from this perspective, it seems to be appropriate that we should give our best to understand what's going on in these sediments. Basically, they are a 1-to-1 mixture, approximately volumetric mixture of particles and water. And the fact that the water can move within the sediment will get really important in a minute. Obviously, these sediments are really important for the global fluxes of materials, and there are different ways to estimate fluxes. A pretty straightforward method to do this are benthic chambers. You basically enclose an area, and you take samples from the overlying water, and by this you can estimate the flux, for example, of oxygen, carbon dioxide, and so on, between the water and the sediment. This obviously typically encloses areas of a few hundred square centimeters, maybe to a square meter or so. Newer technologies like the eddy correlation are really nice to get the flux measurements on larger scales. So basically you have a velocity Doppler instrument and an oxygen microsensor, and by some tricky algorithms you can calculate the oxygen flux, for example, from the seafloor, from the water into the seafloor. And depending on how high you set this apparatus, you get a footprint that can be pretty high. So this is the height of the instrument, and this is the footprint, so basically the area over which you integrate fluxes. And you can see that you can integrate fluxes over, for example, 80 meters. So you can basically measure the flux from an entire seagrass meadow or so on. Now, on a very small scale, the introduction of microsensors have improved our understanding of very much. So in the 80s, sometime, people started to measure oxygen profiles, and from these profiles, so these are 1.5 millimeters, pretty narrow oxygen penetration in these kind of muddy sediments. From the slope of these curves, you can also estimate fluxes, so oxygen flow, diffusive flow into the sediment. If we now go into the sediment, it's getting pretty complex, and most of you, I think, will know that organic material, sediments, and in the sediment there are a whole lot of reactions taking place. People talk of the redox cascade because, well, the deeper you go, different electron acceptors get important. But I'm mostly interested in the oxygen, not only because we have the sensor to measure them, but also because many of these other reduced compounds in the end will get oxidized due to the presence of oxygen. Now in these book-like pictures, you often get something here on the side, bioturbation. So there's something maybe not so right, or maybe we should account for this, but often, well, all this nice layering can be really heavily influenced by bioturbation. That has not always been like this. So there has been the Cambrian substrate revolution. Before 530 million years ago, the seafloor was mostly covered by microbial mats, and only afterwards animals start to burrow into the sediment and open this three-dimensional space for other organisms. So the fact that animals start to burrow, most likely because they needed to hide, because there were so many predators evolving, they burrowed into the sediment and opened this habitat for many other organisms. So they have probably played an important role in this explosion of species during this time age. Today, on average, Bernard Boudreau calculated that the seafloor is bioturbated 10 centimeter deep all around the world. The volume of the bioturbated sediment is probably approximately like the volume of the Great Lakes. And these animals that bioturbate the sediment, they have been there for a long time. These are Jurassic shrimp burrows found in Israel. This is a picture I got recently from a colleague in Canada. This is a Jurassic tidal flood. You see the ripples, you see some polychaete poop here, and it's amazing that it's that nice preserved. So they have been there for a while, and bioturbation, by definition, the simple way to define it is the displacement of solids and fluids by organisms. And there are some models that describe, and people here like Josie were involved in getting these, in producing these concepts of bioturbation. So if you have a physical disturbance, so for example, you put a plastic sheet over the sediment, everything dies in below. So and this is here in time. You will see that in time, animals will burrow deeper. It will go from opportunistic species to deep burrowing animals. Graphs like this you can also have from heavily polluted sediments to pristine sediments, from muddy sediments to more permeable sediments, and so on. Bioturbation in general is a challenge to investigate, but it's also really fun because you have all these different disciplines that you have to deal with, because it's a physical process, but it's driven by animal behavior, and it affects biogeochemistry. It is important for benthic ecology, and can even create these entire landscapes like these thalassinids that you see in the background. I mostly will focus on the movement of fluids today, and I will also show you why. So I told you already that microsensors have been a great tool to get these, to know, for example, that oxygen penetrates just a few millimeters into the sediment. But if you do these profiles every now and then, you get an awkward profiles, which is not like the other ones, but that potentially shows you that there is something going on that has to do with animals. And with lots of work in the 80s, people measured lots of these profiles, and from the profiles, they basically could extrapolate to these two-dimensional maps. Now, we have gone a little further. Now we can use planar optodes. And what that allows you is basically taking approximately 1,200 profiles with one shot. So instead of measuring here and here and here, and well, getting the lines so that it fits with the profiles, you get one image at a shot that shows you 1,200 profiles. And not only that, it allows you to take images every 15 seconds or so. So what we start to see is these tremendous dynamics. So what you see is black is anoxic sediment, the overlying water has some oxygen, and down there an areniculate polychaete is pumping water. And just to show you that it's extremely dynamic, so with microsensors, you potentially cannot go really far. There are two basic models how you can think about the movement of fluids, or the movement of fluids and solutes through the sediment. And one obviously is Bob's classical cylinder model. So in a muddy sediment, for example, imagine a U-shaped burrow, and from there oxygen, for example, diffuses into the sediment based on the concentration gradient. Now, since, well, in the 2000s, Philip, for example, was one person who really started to look more at the advective part of all this. So if you have an animal like an areniculate who lives in a blind-ending burrow, these animals, they need to pump water into the sediment. And what Philip did is he modeled the pressure field that are related to that. So when an animal is pumping water into a burrow or into the sediment, this will create a pressure field, and this pressure field will result in flow. So the basic, really simplified equations to describe the flux here is for the diffusive flux, you have a diffusion coefficient, and based on a concentration gradient, you get a certain flux. For the advective side, for the advective flow, you have another constant, not a constant. This is the permeability, and I will describe this a little closer. And so the flow is basically dependent on the permeability of the sediment and the pressure gradient. So the C here is a constant, which is based on the temperature and the density of seawater and so on. Not so important. So the important ones are these two: permeability, pressure gradient. If you have a large pressure gradient at a certain permeability, you get a high flow rate or a large flux. The velocities of these flows, for example, of oxygen, are really different based on the environment we are looking at. So the diffusive flux, an oxygen molecule, may move about a centimeter in 10 hours. With advection, animals pumping water into the sediment, 1 centimeter, an oxygen molecule may move 1 centimeter in less than a minute. So in these advective systems, the flow of constituents, of pore water constituents, can be extremely high. Now, permeability K is a parameter that you can measure, and it describes how easy the ease of water to flow through sediment. So if you have a permeable sediment, a coarse sand, water easily flows through the sediment. And as you go to more muddy sediment, the water will not flow that easy. And you can just measure it by putting a pressure head over a core and measure how easy water flows through it. Now the interesting thing is, if you look for the sediments we are looking at, so for example from muddy sand to pretty beach kind of sands, this spans a permeability of about 2 orders of magnitude. And if you now look back, that the flow rate or the flux here is related to the permeability and the pressure gradient, that means in a sediment that has a 100-fold permeability, the animal would need to induce a 100-fold pressure to pump water into it. So it's also really interesting or important from the energetic standpoint of the animals. Imagine a really simple experiment. You have an aquarium or whatever. You have a head tank that you connect to an inlet. You have a pressure sensor that is connected to all this tubing. And if you raise the head tank, you will increase the source pressure with which you push the water into the sediment. And at the same time, you can measure the flow rate. So the water drops that come out, that move up, and then move out of the aquarium. And what you get, because physics typically work, is a linear relationship of the source pressure versus the flow rate. So the further you increase the source pressure that you measure here, but that you arrange with a head tank basically, if you double the source pressure, you will double the flow rate. Pretty simple. All that depends on the hydraulic conductivity. So if you have a 5-fold hydraulic conductivity, for the same source pressure, you will get the 5-fold flow rate. We not only induce the source, we can not only use the source pressure, because once we inject water into the sediment, we pressurize the pore water. So Sally and David, they started to work with pore water pressure sensors. So basically you connect the pressure sensors with the pore water, and then you can also measure that, the pressure in the pore water. So for this kind of sediment, for example, you will always have, well, if you measure these source pressures, you will get somehow lower values of pore water pressure that you can measure at the same time. And with these pore water pressure sensors, basically, we stick it into sediments where we have animals, and so we get the pore water fluctuations that are induced by these animals. Now, the relation between pressure and flow rate has also been investigated by many other people before. So, for example, Foster Smith or Riska, they estimated these relations. So, if we just look for an arenicolate, for example, they pump a maximum at a maximum rate of about 50 mils per hour, and they can pump. They know it from having a worm in a glass tube, and then they raise the head pressure in front of the worm. They can pump up to 20 centimeters of water. So that's the pressure unit in this case. Now, if you look at this arrangement here with a maximum pressure of 20, you could pump in this kind of hydraulic conductivity. You could pump 100 mils per hour. So this animal would already be at its edge in this case. We know that lugworms occur at lower permeabilities, and in a moment I will show you why we think why. But this is already important to remember, that the pressure and the flow rate, this ratio, is also really important from an animal perspective. So Sally and Philip modeled these pressure gradients. Sally and David got out, okay, let's put pressure sensors into the sediment and measure what we get there. So this is False Bay. It's a really nice place because you have phallocynids, you have tellinids, so bivalves, but you also have areniculates. And so areniculates are these worms that, for example, live in blind ending burrows. They pump water into the sediment. They do this by, well, here speeded up peristalsis. So these packages of the worm that press always about 0.2 mils per peristalsis, and they do this 5 or 6 times per minute. So they pump this water into the sediment. You see this nice oxygenated halo surrounding the worm. And if we put now pressure sensors into the sediment, this is what we get. So we basically get all these different behaviors. We get a nice pore water pressure waveform. So all these small fluctuations, it's each of these is one peristalsis. Every now and then, they need to defecate. It's a large, big negative signal because about 2 mils of sand are pushed out through the tail shaft onto the sediment surface, which causes a large negative pulse. And also burrowing typically induces large oscillations around this baseline here, around this hydrostatic baseline. So from these pressure signals, we already know that these animals, mostly they pump water into the sediment. That's what they should do because they need the oxygen. So that's basically why they are pumping. But every now and then we see these negative pressure signals. So for short periods, and actually for lugworms, we know that it also can occur for 10 to 15 minutes long. They reverse the flow and they push water out of their burrows. So in our work, some of the main questions that we had over the last 4 years are, who are the big players? So we know this now for lugworms, but who other animals, or which are the other species that may be important in terms of pore water fluctuations? And how is all this related to the functioning of benthic systems? So when we started our work, I was basically working here at the Waddensee station, and this is how the tidal flats there look for hundreds of kilometers. So well, it's not a monoculture of lugworms because there are other species living in these tidal sands. But as you can see, these intertidal flats are really dominated by this polychaete worm. And my PhD was basically to find out, well, what do they do for the ecosystem? And an easy way to find out is to get rid of them, make an exclusion experiment. And I did this on a pretty large scale. So down there it's me. I hired a guy with a backhoe. He took away the upper 10 centimeters of sediment. I rolled out like a carpet, a mesh, like a fly gitter, 1 millimeter size. And I did this on 20 by 20 meter areas, 6 times replicated, and I got these nice square 20 by 20 meter exclusion plots. And over the next years, I investigated what animals do settle there, how does the sediment change, and so on. These plots are still there. Luckily, you can even see them on Google Earth, Google Maps. So they made a nice mapping for us. So these black plots are here, the exclusion plot. Actually, there are 2 more. In the beginning, I used a wrong mesh, so that's why there are 8. But basically, there are six plots of these. And I don't want to go in detail here, just to tell you that the absence of lugworms, they really had dramatic consequences for what was going on, because in the absence of lugworms, these sediments, they got muddy, poor water nutrients increased. There were some animals that enjoyed not being disturbed by the bioturbation of lugworms. Others, they could not live there because there was, for example, so much sulfite. But mostly why I told you about this experiment is because we used this experiment, and Sunny and David, they came over and they heard about this experiment. We used this experiment to put pressure sensors in an array from the lugworm populated site into the exclusion plots. And by this, we could basically estimate the pressure wave propagation through the sediment. So this here, for example, is a diffication, and the lines here correspond to different pressure sensors with increasing distance. So diffication, which is frankly the largest signal of the animals, but still we could detect 45 centimeters away from the animal. So an animal like this, 45 centimeters away, we can detect a diffication. Now, if you think of a tidal flat like this, where nearest neighbors are around or below 10 centimeters, you can imagine that, well, the entire water is basically moving by the activity of these organisms. And it's not only during the diffication, it's also during the peristalsis, even though the area of influence is then a little smaller because the pressure signals are not that large. So to link these pressure dynamics and what we know from the pressure dynamics about advection, about pore water flow to the geochemical impact, I started to work with planar optodes. Lubos Polaretsky, he at the MPI developed this technique with some other people, and since then we have used these pink optodes, which are oxygen sensitive foil, with a fancy camera, which you can see here. You can detect the quenching of fluorescence in the presence of oxygen. And these are probably, this is David showing proudly the largest planar oxygen optodes that have ever been used. These are 50 by 50 centimeter for thermocentric shrimp. So this is how typically our setup looks like. We have the optode camera here, some aquaria, pressure sensors in there, camera on top. So we try to get these data synchronous for these animals to link behavior and biogeochemical impact. So some first examples I want to give you for another animal that produces these mounds, which are thalassinid shrimp. You can find them, well, almost globally, especially in the tropics. They can be really abundant, but also in the temperate regions. So this is a picture from Oregon. This is Neotropia californiensis, and well, it's a shrimp that gets 10 centimeters long maybe. It builds quite complex burrows, moves around. This is an egg-carrying female. And once they have established a burrow, this is typically how they irrigate the burrow. With play-put beads in the narrow parts of the burrow, they sit in there, and this is how they pump water through the burrow. So with each of these pumping events, at a certain point within the burrow, we get these rises in pore water pressure. And when I start this movie now, you will overwhelmed by information. Nevertheless, I will show it. The shrimp has this burrow, which is here about 40 centimeters deep. It will move up and down. This is the synchronous pressure record and the synchronous oxygen dynamics. Speed it up a little bit. You see, the shrimp is moving down all the time. You see different pressure plateaus depending on how close the shrimp irrigates to this pressure sensor that is shown here, and you see the related oxygen dynamics here in this slide. In the next video, I try to slow it down a little bit more. So the shrimp is now sitting here, irrigating in this direction, and this corresponds to this pressure plateau. Then it will move to a different location. I think where we cannot see it, that's obviously always a little sad, but this is nice about the pressure sensor, because even though you don't see it, you see that the animal is irrigating because we get these pressure plateaus. And corresponding to this, so now it's here, well, now it's again somewhere where we don't see it, still something, but you still can see that, well, with the normal imagery, we could not get all the information that we can get from especially the pressure sensors and the relation to oxygen dynamics. And the oxygen distribution is really dynamic. So if you look at these time profiles, profiles in time, so this profile here is shown here in time, so over 24 hours, you see that these oxygen spheres surrounding the chambers is growing and shrinking, growing and shrinking. You see some synchronous things going on here because probably these two parts of the burrows were always affected by the same locations where the shrimp was active, a little asynchronous when we look down here because, well, typically the shrimp pumps water in this part of the burrow, then it goes down, pumps the oxygen further down, and so on. So from these, and well, this is not terrifically new that oxygen distribution, for example, is very dynamic in these sediments. But I think what is really important and what we try now to do is giving this dynamic some numbers. So you just saw these distributional maps. Now we look at these two pixels here. Imagine, well, the pixels are smaller, but imagine that these are the two pixels and we have these oxygen time series. So oxygen concentration here on the y-axis, the x-axis is time. This is maybe, I don't know, an hour or so. So you see all these ups and downs in oxygen. And let's just focus on the second series here. So you see that there is a certain amount of time where there's no oxygen, then there's oxygen, then oxygen is consumed, and so on and so on. So from these series, for each of these pixels, we can basically calculate the duration of an oxic event, and we can average this over all the oxic events that occur. We can define the duration of an anoxic event and average this over all these events that will follow. And from this, from the relative time of oxygen being there and oxygen not being there, we can estimate the probability of oxygen. We can also estimate the number of oscillations between oxic and anoxic conditions per hour, for example. And we can calculate the average oxygen concentration. So whenever oxygen is there, what is the average oxygen concentrations? And we end up with maps like this. So this is this burrow chamber deep down in the sediment. So there's in the center of the burrow, oxygen is there for a long time, for long time periods. It's surrounded by a sphere that is highly oscillatory. Oxygen concentration during this short time was pretty low. Now we get then maps from the entire burrow. So this is a 50 by 50 centimeter end farm. This is the approximately position of the shrimp burrow that you cannot really see. You see some chambers here, but it's really hard to see. So what you see here are the oxygen probabilities. So within the burrow, they are close to 100. The burrow lumen is mostly oxic all the time. But the sediment surrounding the burrow, so it's oxygenated most of the time. That's why in this map of the redox oscillation, it's 0 inside the burrow, because there are no oscillations between oxic and anoxic condition, because it's always oxic. But surrounding the burrow, you have these, you have the sediment that experience very frequent oscillations. So on the scale of 1.5 times per hour. The oxygen concentrations are typically below 30 to 40%. Typically, for all the animals we tested, below 50%. That's logically because the animals need some oxygen for their respiration, so they take away already a way big part of the oxygen. And well, on its way down to the burrow, there's already some consumption. The duration of anoxic events is typically longer than 1 hour in the center of the burrow. But well, again, surrounding the burrow, there are many. There's a large sediment volume that experience anoxic conditions for, well, let's say the green parts here, half an hour. So there's oxygen, and then every now and then, half an hour, there's no oxygen. So we get a much better idea about these numbers. And we can put profiles through these maps. So here's, for example, again, a map through this chamber, which I showed you a little earlier. So you see the red, and maybe we just should focus on this. The red color shows you the number of oscillations So in the middle of in the center of the chamber, there are not so many oscillations, but as soon as you go into the sediment surrounding the burrow, you have lots of oscillations, simply because the oxygen consumes the sediment very fast. So whenever the shrimp stops irrigating, oxygen is consumed and you get an anoxic event for some time. Interestingly, and because the animals pressurize the sediment over extended distances, so let's say for the sediment maybe 10, 20 centimeters, you also see these oscillations at the sediment surface. So if you put this really tiny profile, which are however about 2 centimeters, through the sediment surface, you get the same picture. So basically it's not about a fixed profile that you would measure with a microsensor. It's all about oxygen probabilities and oscillations between anoxic and oxic conditions. So in this case, the diffusive boundary layer is basically an oscillatory boundary layer. And this may have important implications. So for example, Peter Stief from the Max Planck, he investigated what is happening in the sediment while it's passing some, well, in this case it was some insect larvae, but in the meantime he has done this for many other benthic species. And so imagine these animals, they swallow surface sediment, there's lots of nitrate in the sediment, but once it comes into the gut, it's getting anoxic. And this is exactly where denitrifying bacteria can get active. And well, typically denitrification would be this steps, would occur in these steps of reactions. Now, in these animals, the sediment is only in them for half an hour or so. For many bacteria, this is not long enough to fulfill this entire pathway. And that's why you can see with an increasing density of these insect larvae, you see an extremely high release of N2O, which is basically an intermediate product. So due to these oscillatory conditions, it's not only that the rates will change, but likely also the quality. And what happens here inside the animals, anoxic conditions for half an hour, and then you are up on the surface again. That is also true for large volumes surrounding burrows. So, well, the concept behind this is basically, this is what we use to measure. It's an oxygen profile, the oxygen concentration. Now, in reality, with these hydraulic active sediments, organisms, we basically should think about oxygen probability curves, which span a much larger height through the sediment-water interface, where the duration, the average duration of an oxic event, well, obviously is totally oxic up here, but because anoxic water is pushed upwards, their oxygen probabilities are most likely lower than 1, close to the sediment surface, and they decrease with depth. The opposite is true, obviously, for the anoxic time. So down here is always anoxic, but because water is pushed up and down through the sediment-water interface, the duration of anoxic events will decrease when you go up through the sediment. And where both T-ox, the average duration of oxic, and the average duration of anoxic events are both short, there you will find this maximum of redox oscillation, or oscillations between oxic and anoxic conditions. So the oscillatory boundary layer is much thicker than what you would expect from a static profile, and the thickness of this layer is most likely dependent on the sediment permeability and the reactivity of the sediment. So in a really high oxygen consuming sediment, low in permeability, the zone is probably really thin, as the oxygen penetration would be, but in higher permeability this can be 2-3 centimeters thick. While the temporal pattern, so how long is T-ox, how long is T-anox, how often does it oscillate between these different conditions, is mostly dependent on the activity of the organisms. To address this, or to now do the next step and address questions like, so how does that affect microbial activity, we developed, well, this nice animal, which is the Robolac, which allows us, and you can see, this is the pressure record from Robolac, and it's much more beautiful than from a real animal, because you can really, well, obviously it's not changing because it's always at the same position, and you can also really change whatever you like. So you can stop the activity, you can pump slowly, you can pump harder, you can put some spikes there and so on. And down here are again, well, these two profiles in time, and we basically really can induce the same oscillatory conditions that we see with the animals, but we can repeat them. We can do it at the same time in several aquaria, and so do real replications, which can be sometimes tricky with real animals. Going back to this large-scale exclusion experiment and thinking again about the spatial extent of how these animals affect things, it's not surprising that in this experiment I found, for example, if I do profiles going from the lugworm exclusion plot out into the arenicola populated side, you see this, well, here's the mesh, and so these are silicate concentrations in the pore water. There are high concentrations below the mesh on the exclusion side, while where you have lugworms, all these nutrients are flushed out, or at least they are transported upwards. So the consequence of this may be, and that's what we observed, that, for example, silicate, which is an important molecule for Microphytobenthus, is if these nutrients are pushed upwards all the time, then this may explain this pattern here. So again, it's the border of the exclusion side here to the arenicola side, and sometimes I saw these huge algae blooms on the surface, but here just in the presence of lugworms. So may that be that this is due to that? So we did some experiments on that. This here is RoboLug. So peristaltic pumps, many tubes going through these buckets, and well, they inject water into these buckets at about 15 centimeter depth, or exactly at 15 centimeter depth. So that's what the lugworm would do. And then we use another really nice camera, which is here, which is a hyperspectral camera, which scans the surface and gets the spectral information for each pixel, which means you can basically estimate chlorophyll from it if you look for the absorption spikes of chlorophyll. So we run this for a few days, and you see there's a nice microphytobenthic community developed due to the oxygen bubbles. There's this cold spot in the middle. We still don't know why that evolved, because that was true for all the flow treatments. But more importantly now is, well, when we irrigate for, I think this is after the second day, irrigate with 1.5 mils per minute over 2 days, you see these chlorophyll maps, which are much higher if we would not irrigate. Which means, yes, this advection of pore water and the advection of nutrients in the pore water has potentially important implications for the primary productivity of these sediments, which is nice for the worms again, because they feed on these surface sediments that they subduct to depth. Now, with 1.5 mils per minute, I just want to make clear, this is like drop, drop, drop. So it's not much water. It's really just really little water flow through the sediment, but in the end it has this huge effect. So another species that may have some kind of the same effects make these traces, Telainid bivalves. This is Macoma nasuda from Friday Harbor. They have a long in-current siphon, a pretty short excurrent siphon. That's why the excurrent siphon typically stays below the sediment surface. So whenever they inject water and surface sediment, they they push water into the sediment. This is here a video where you can see the in-current siphon. It moves around, feeds here and there. Every now and then you see it moving. Well, there's a cough. And every now and then it moves like a mole through the sediment, goes out here, feeds there. And this is how it moves around, while the excurrent siphon, which you see here, you see all these fecal pellets here, you can see this movement of water through the sediment. So whenever the animal is feeding here, here's the in-current siphon, now it will cough. Then you see that water is injected into the sediment down here. So this video again, this is the New Zealand species, it burrows, you see these huge signals, and this animal was really cooperative, but because at one point it puts its excurrent siphon somewhere here. And once that happened, you see that with each of these pressurized plateaus, you see an oxygen bloom here. So here, pressurized. Some burrowing, pressurized. Some burrowing, again, pressurization, stop. Pressurization, stop. So here it's again an example how nice pore water pressure signals correspond to the oxygen dynamics. The nice thing about the pressure sensors is that we can also put them in the field and confirm that this is what they do also in the field. Now I talked about this potential gardening effect of these animals, and this is a really nice video. So this is 25, so these are basically 3 days in a few seconds. the in-current siphon is moving around. This is where the ex-current siphon is. You see a brownish layer, which is the microphytobenthos. Here it's irrigating, and on the third day it stopped irrigating here, and now you see what happens. So luckily it did not feed here again, but you see how within a day there was a huge growth of microphytobenthos exactly in this position. That may not only be due to the previous upwelling of pore water, but also to all these fecal pellets that were deposited below this zone. The last thing I want to emphasize here, these are two videos of lugworms. Again, the J-shaped burrow, but two different permeabilities. So basically the animal does the same, but you see a qualitative, totally different picture here. While on the left side, it's more like a sheet flow in this permeable sediment where water easily can flow. In this low permeable sediment, well, you see all these little plumes coming out whenever the animal is active. You don't see very much going on here because it's a high oxygen consuming sediment, so oxygen does not come to the foil. But you see that, well, how the anoxic or how the water is pressed out of the sediment. And so why is it so different here? And does the animal do something about it? Now going back to Darcy's law, which was pressure gradient, flow rate, you can run a really simple experiment. Basically, an inlet down here. Behind there, I'm raising the head tank step by step. I measure how high I am. At one point, you will read a critical pressure, and you will crack the sediment. Now, this is a gigantic crack. But already before, you can see that there is a change. So this is, again, the source pressure, so the height of the head tank, basically, and the flow rate that I measure. So for a long time, it's pretty linear. Then it somehow deviates from the linearity that it should have. And then here at 25, I produce this large quicksand column, which then in turn really changes the flow rates that I would get afterwards because there was lots of sediment sorting and so on. But the most interesting thing I think is this here, that even, well, Darcy's law should still somehow work, but already there is a deviation from what we would expect just from simple physics. So one experiment that we ran is with the optodes together. So we irrigate with 1 ml per minute, which is approximately what a worm does. We measure the source pressure and the pore water pressure. And we do this with a constant rate. And then in the second experiment, or in the same experiment, we add to this continuous rate, we add these pulses, which are produced by the same amount of water that we see for these lugworm burrowing spikes. So this is 1 ml per minute. Pretty stable, everything is nice. Or some plumes, it's low permeable, but now we add the spikes. You see some flashes here, and you see how some of these small cracks really open here at the sediment surface. So yes, that really suggests that these animals are capable of cracking the sediment hydraulically, which in turn may have, again, really important implications for microphytobenthos, because, well, we also did not only use the hyperspectral camera in the lab, like in the small buckets I showed you before. This is a scan over an arenicola tidal flat. And you see this, again, a map of chlorophyll distribution. And you see this enormous heterogeneous distribution of chlorophyll at the sediment surface. So the lugworm poops are dark blue. There's hardly any chlorophyll in there because the animal digested this. But beside of that, you sometimes see these hotspots of microphytobenthos, and these may also be due to the fact that these animals have introduced a crack to the surface where all the time all these nutrients are upwelling. Nice place for microphytobenthos to grow. So in summary, bidirectional, which means forward and sometimes backward, pumping by these animals cause pore water advection, and this seems to be really a prevalent feature of biogenically active sediments and, well, in a strict sense, in permeable sediments. So, but in permeable sediments, I mean also muddy sands. So it's not like only coarse sands, it's also pretty muddy sands. In real muds, well, we won't see these things because these sands do not allow pore water to flow through them. But in permeable sediment, in the presence of such animals, this is a really important transport processes. And as some of the consequences I showed today were these redox oscillations, and one thing I would really like to do in the future, look much more into how do these oscillations affect microbial activity and diversity, for example, the degradation of organic pollutants in these sediments and so on. The upwelling of pore water, or that these dense beds of organisms, that these cause some emergent properties, Sally told me, so that they act like a bed. So the entire bed, Arenicola flat, for example, is fertilized by these mostly upwelling pore water all the time, which obviously has important implications for the productivity of the system. And then there are interesting implications of this hydraulic cracking in different sediment types, and this has implication for the energetics. I forget to show you that after these cracks occur, the source pressure was 25% lower, which means after that it's 25% easier for the animal to pump with the same rate. So all this has important implications for the energetics of the animals. So going back to the concept of organisms living in sediments, going from disturbed to undisturbed areas, or well, time after disturbance, or from polluted to unpolluted site, I would like to add this fourth dimension to it. So these are maps of all kind of animals, and in a moment I will go a little bit more in detail as my last slides. But so this is going from really muddy sediments to very permeable sediments, and these are well, here for example the oxygen probabilities for these different animals. Well, these are all assembled obviously from ant farms and aquaria measurements. We need to think about oxygen oxygen oscillations, and that oxic versus anoxic conditions only last for a certain amount of time. And so how do microbes deal with these environments? Because this is what they experience. So in this slide, in this animation, you will see all kinds. There are some meldanids here. Macoma, so thelinids especially, they produce redox oscillations of up to 6 per hour. So really frequent oscillations. Some deeper thalassinid burrows. This is the one I showed you several times before. Another neotropia and a little bit more permeable sediment. Some arenicolids here. Down here is always, well, we're going from low to high permeable sediments. Especially going to the higher permeable sediments, the areas that are affected by these oscillation conditions is increasing. So I forget to mention, this is the scale going from 0 to 4 per hour oscillations, toxic, anoxic oscillations. Another lugworm here, an Ares, which also causes here an advective zone. And so, well, I think this is really nice that we now have a much better idea about the numbers that are related to these oscillatory conditions. So over the last years, we worked in many different labs to get access to all these animals, to get access to all this cool information that people have about these different species. At USC, we work with some microbiologists. At the MPI, Arjun is doing this hyperspectral imaging mainly. Ted DeWitt is the thalassinid guy, Carsten Justus, lugworm people, Simon Conrad, Judy, they are thellinid guys, Alf Joanna, also thellinid, and Maricellaria, and so on. I want to thank all of them. I want to thank the funding agency, especially NSF, who has funded my postdoctoral work over the last 3 years. And again, you for inviting me, and I'm happy to answer questions if there are.
There must be some questions. Alvin.
How hard do the worms have to work to keep the tunnel open?